Hello everyone! Today I'm going to present my Bio 110 Ambassador Program Project Mitosis. We typically use onion roots as the experimental material to observe mitosis because we can look at the root tips. A portion of the cells at the root tip belongs to the meristematic region, which is ideal for observing cell division. Let's briefly review some information about cell division. The familiar M phase is mitosis, which is divided into prophase, metaphase, anaphase, and telophase. The remaining phase are interphase, such as G1 cell growth for DNA synthesis, S-phase DNA replication, and G2 preparation for mitosis. This is the cell cycle analysis process. We can take a closer look at images of each stage of cell division, which helps us better observe different cells during the experiment. Prophase, the nucleus becomes visibly enlarged. Metaphase, chromosomes align neatly along the cell plate. Anaphase, centromeres separate and chromosomes move apart. Telophase, two new cells are formed. Here are some details of the experiment. We take cells from the onion root tips. It's important to collect about 1 centimeter of root tip to ensure we get cells from the meristematic region. Then we place the cells into a fixative solution to fix them. After that, we add a few drops of hydrochloric acid solution onto the onion root tips and wait for about 5 minutes. Next, we rinse the root tips 2 to 3 times with clean water. Then, we stain them with gentian violet for 3 minutes. And then place them on a slide for observation. We can use the eraser end of a pencil to gently press the root tip, which helps to squash the cells for better observation. In normal cells, mitosis is tightly controlled by checkpoints like the G1S and G2M checkpoints, which make sure DNA is correctly replicated and chromosomes are properly separated. However, in cancer cells, these checkpoints are often bypassed, leading to errors in DNA replication and accumulation of genetic mutations. At the same time, cancer cells are prone to chromosomal instability during division, including misalignment, breakage, or aneuploidy. This instability not only allows more mutation, but also drives tumor progression and malignancy to accumulate. As a result, regulation and chromosomal instability are key mechanisms behind cancer development and growth. Next, let's talk about some drugs that inhibit mitosis. The first major category is CDK inhibitors, cyclin-dependent kinase inhibitors. Drugs in this group, such as palbociclib and ribociclib, specifically target the key enzymes CDK4/6 which drives cells through the G1S checkpoint. Under normal conditions, cells pause at this checkpoint for a self-inspection to ensure that their DNA is ready for replication in the S phase. However, many cancer cells bypass this safety mechanism by overactivating CDK4/6. CDK inhibitors block these enzymes, forcing cancer cells to remain in the G1 phase, preventing DNA replication and thereby effectively slowing or stopping tumor proliferation. The second important category is microtubule inhibitors, such as paclitaxel and vincristine. These drugs directly disrupt the spindle apparatus, a structure essential for mitosis. The spindle is responsible for pulling apart duplicated chromosomes and distributing them into two daughter cells. If microtubules cannot properly polymerize or the spindle cannot form or remain stable, leading to chaotic chromosome segregation. Cancer cells are unable to push through this stage and often trigger apoptosis. Therefore, microtubule inhibitors are widely used in the treatment of many types of cancer.